everyone, my name is Timothy Ayers, and today I'm here at Berkeley's campus with Dr. Dr. Scheckman won the Nobel Prize in Physiology and is a cell biologist here at UC Berkeley. So Dr. Scheckman, I've already introduced you just a little bit, but I'd like to ask, who are you and what do you do?
Again, Randy Schekman. I'm a professor of molecular and cell biology at UC Berkeley, and this is the middle of my 47th year here on the faculty. Although I was born and raised in Minneapolis, Minnesota, we moved to California when I was 10, Southern California, and I went to UCLA in the late 1960s as an undergraduate, had a great time, and became really captivated by science, particularly by biology, life science. So I did well enough to be admitted to a terrific biochemistry department at Stanford, where I was a graduate student in the early 1970s. And then after another period of study in Southern California, I took the position here at Berkeley, where I teach undergraduates primarily. I teach introductory biology this semester, and I also do research on cells, in particular on cell membranes and how cells put these membranes together.
So tell us about your early childhood and how you found yourself even thinking about pursuing a career in science, less more winning a Nobel Prize in Physiology.
Well, that of course wasn't on my mind when I became interested in science, but I do remember my earliest interest came from a toy microscope that I got probably when I was 11. And I remember I lived in Orange County, which was much more suburban, almost rural than it is now. And there was a dry riverbed near our home, and I collected a jar of pond scum. Don't ask me why I did that. But I took a drop of this film on this pond scum and put it on a glass slide and looked into this toy and saw this amazing world of microorganisms crawling and swimming around. And I was, for days after, I was endlessly fascinated by this. And I saved my money, and I eventually got a student professional microscope, and that became my pride and joy through the high school years, and where I every year entered a science fair project and got recognition for the work that I'd done independently. And this was, for me, the way that I became interested in research. I started at UCLA with the intention of going to medical school. That seemed to be the only path that I might have given my interest, but I realized very quickly when I was working in a laboratory in my freshman year that there was another path, and that was to do science and do research at the cutting edge. And that ended any interest I had in medicine, and I have pursued that ever since.
I see. So what's your hot take?
Oh, well, there are many things, but I think for this audience, one thing that I would like to bring to the discussion is the power of science, the power of logic, and the way that scientists work. It's not widely appreciated that science is not a process of coming to some absolute truth. It's a process of discovery that may approach a truth, but never actually reaches the truth. It's virtually impossible to prove something. You can develop good evidence for it, like we have terrific evidence for evolution. Evolution is a powerful force, but there continue to be refinements in the theory of evolution. And people don't seem to understand when they say it, when we call it a theory, that makes them think that it's not a certain thing. But the evidence for evolution is overwhelming. And so it remains a theory because there are parts of it that still require further study and refinement. So the Nobel Prize in Physiology this year was given for somebody who sequenced the genome of Neanderthals. And the idea that you could actually do that 20 years ago would have been foolhardy. That you could get enough DNA from a sample of Neanderthals who died out 30,000 years ago to actually be able to sequence the entire genome. It was seen as virtually impossible. But with technology and advances in the procedures that we use to sequence genes, it became possible. And this fellow, Svante Pääbo, who was studied here at Berkeley when he was a postdoctoral fellow, has led the field and made this tremendous advance. So science has boundaries that keep pushing forward, and that's what really makes it so exciting.
So in words for a non-scientist to understand, what is a secretory vesicle, and how did that become incorporated into you winning the Nobel Prize in Physiology?
Yeah. Okay, so the cells in our body have all the chromosomes. Most of the cells have all the chromosomes housed in the nucleus, which is in a cell. And it's about 23,000 genes. The human genome has about 23,000 genes, which roughly means 23,000 different protein molecules that are coded by these genes. Now, all of these protein molecules are made from instructions from the nucleus, are made inside the cell in the part of the cell called the cytoplasm. And so you've got all these proteins being made and bouncing around. But some of them, a good fraction of them, maybe a quarter of them, end up being exported outside of the cell. It means they have to breach the barrier that surrounds the cell, which is a biological membrane. And they don't just punch holes in the biological membrane. These proteins that are destined for export have to be encapsulated by recognition of some special aspect of their structure. And so they become encapsulated eventually into little membrane carriers called vesicles. Mm-hmm. And these vesicles are little packages, they're shipping packages that deliver the proteins like insulin or antibody molecules that we use, deliver them to the perimeter of the cell where the vesicle merges with the membrane that surrounds the cell, and what was inside the vesicle becomes the outside of the cell. So that's how insulin is secreted by the beta cell of the pancreas. And then insulin diffuses around, it goes into the bloodstream, and it is taken up by other cells to do its thing. And there are thousands of different proteins that have that path of transport. Now, this was known at a superficial level in cells that were looked at in the microscope when I started in the mid-1970s. But what was not known was how this process is operated. What are the nuts and bolts that actually manufacture these vesicles and move them in cells and allow them to merge so that what they're carrying can be exported from the cell. So what I decided to do was to study this in a simple microorganism. Again, my interest has always been in microorganisms. So it turns out that there are some microorganisms that are called unicellular eukaryotes. So we're a eukaryote. There are a lot of eukaryotes in the world. All of them are organisms that have a nucleus, as opposed to bacteria, which have a chromosome, but they don't have a nucleus. And so it turns out baker's yeast, which we all know and love because of what it does, is a unicellular eukaryote, which means that the machinery that a yeast cell has is very similar to the machinery that's in our cells. The machinery that allows a yeast cell to operate, and which we've borrowed, evolved on Earth maybe 2 billion years ago. And the nuts and bolts of that machinery have been passed along over all that time so that they operate in more or less the same way in our bodies. Now, this was beginning to be appreciated when I started my work. And so I decided, for technical reasons, it's much easier to study this whole process in a yeast cell than to have to work on a human being or even a cell from a human being. It's really much simpler to grow microorganisms and study them, especially if you want to use an approach called genetics, where you look for mutations in these genes that cripple a process. Much easier for technical reasons to do this in yeast than in human cells. And so over the course of the first couple of years I was an assistant professor, we isolated mutations and defined a couple of dozen different genes. We now know there are many more such genes that are required for this, that code the machinery that allows cells to export molecules like what a yeast cell needs. But since the genes that we discovered turn out to be virtually the same as the genes in the human genome that do the same thing, it became apparent that the biotechnology industry, which was just developing, could use yeast as a production platform for the manufacture of proteins that would be of clinical benefit. So, for instance, I helped the local company to engineer the gene for human insulin in yeast. And although yeast cells, of course, don't have insulin, they have no need for insulin, they can be fooled into piggybacking insulin along with their own proteins that get secreted. And so now 1/3 of the world's supply of human recombinant insulin that's used to treat diabetic patients is manufactured in giant fermentation vats growing yeast cells, which secrete the human insulin, a recombinant form of human insulin, into the growth medium in these vats, and that can be easily isolated and used for therapeutic purposes. So there are many such examples. And the take-home lesson from all this is that if you make a fundamental discovery just by logic and experiment and theory, there will be a practical application for it. You can be virtually certain. It may come quickly, as in my case, or it may take decades to realize, such as from quantum theory, that there would be applications in telecommunications that couldn't have been predicted in the 1920s, but we all benefit from it, and that was because of a fundamental discovery that could then be applied practically. So that's really, there's a virtue to science just in understanding the living world and in understanding the universe, but it's also because it inevitably has some benefit for humankind.
I've interviewed a lot of scientists just across my stay here in California. And it's been like 10 to 20 scientists. And one of the most common things that they say that really supports what you're saying is, sometimes you have to go against the grain. You can't just do somebody else's experiment. Like you have to try it out, and you have to be willing to fail.
Yeah.
And when you do fail, like what's your method of maybe getting over that, or do you try something new? How do you deal with that?
Yeah. Well, if you train as a research scientist, you get used to constant failure. Most experiments don't work for reasons that you can't predict, or sometimes the failure is instructive and it teaches you what to do right. And so my advice to people who don't necessarily, most people are not willing to take that kind of risk. Most people are willing to work hard and to apply themselves and then achieve some benefit from that hard work. But to be an effective research scientist, you have to be a gambler. You really do have to take risks. And of course, risk implies failure. There will be failure. So the important thing is, when you take that risk, it should be a big, worthwhile goal. It shouldn't just be a little thing. You want to do something big. You want to do something different. You want to make a contribution. So think of the big picture. Devise a plan to develop an understanding of the problem that you've set for yourself, and then keep blinders on and focus on the big picture. Focus on the goal and not to be diverted by other things that may occur to you. Just keep a blinder on and focus on the problem. And if you encounter a failure, then you turn the problem around in your mind and you look at it in another direction, but you just keep going. And in my case, I had been successful before in a very different research setting, a different research problem. And as a result of that success, I had developed a sense of confidence in my judgment. And when I made this decision to work on yeast and to study how protein molecules might be exported, I had no experience with yeast or with genetics or with protein secretion. But I had confidence from my past work that I could formulate a problem and see it through to the end. And of course, there was a lot of skepticism, which was a good thing, because people didn't think yeast would be at all relevant to humans, at least in this field, because the people who'd studied this problem before had always worked with animals. They'd never worked with yeast. And so there was a lot of skepticism. But I had faith, again, my interest in microorganisms, that there was some truth that could be obtained, and in fairly short order it worked well, and then the skepticism melted away.
I see. So with all that skepticism, like a normal person would be like, okay, well, I'm getting somebody from this year and this year telling me, hey, don't do this, Randy, don't do this, Randy. So what made you say, okay, I'm gonna do this? I'm staying focused. This is what's gonna happen.
Yeah. Well, I thought that I didn't think it was as risky as other people thought. Yeast had already been used by other scientists to study problems that were of relevance to human cells. And yeast had for decades been used as a genetic organism to discover new genes. And if you look inside of a yeast cell using a microscope, a special electron microscope, you can see evidence of the kind of machinery. It looks different, but the kind of machinery that a human cell has that others had studied. So I didn't think it was that much of a leap. And given the power of the genetic approach, which really could only be done with a microorganism and not with humans at the time, I felt I had a leg up on any competition that might have tried to approach the problem to solve the mechanism, but using a different approach. And it worked quite well. Now, of course, this all depended on being here at a great place like Berkeley with fabulous students, and I've had the real honor and pleasure of having some brilliant students in my lab who shared my vision and had a strong work ethic and made it happen.
A man once told me that a lot of times it's better to really think through things and, you know, not be so impulsive and really just get it right and do it the right way because you're going to save time. But then another man told me that paralysis by analysis. Yeah. You got to start thinking. You got to start taking action. Yeah. What end on the spectrum do you see yourself
Well, I would
on?
favor what the second guy said, because some people are so smart that they can talk themselves out of any idea or any experiment. So you can't overthink things. Otherwise, you'll never make any progress. And so sometimes it's good to have intuition rather than just deep intelligence. And so, of course, there are plenty of people who are brilliant and do well, but there are plenty of people who have an average intellect, but who have a special gift, a special intuition that allows them to make discoveries that the even smarter people couldn't make. So I fully agree. You know, you have a good idea, you plan it out, and then you go and do it, and don't waste time overthinking the problem.
I gotcha.
Because it's by the act of doing and learning from experiment or from theoretical considerations, it's through that act that you refine the idea and approach the truth in a way that you can't just necessarily imagine without having done a little experimental work.
So somebody once told me you're 1 in 5 of everybody you hang around. Is that true?
1 in 5 of in what?
So everybody, this friend is 20% of you, and you're 20%, and 20%, and then 20%, and then 20%.
I see. Well, it's science is a community activity. Very few scientists succeed on their own. Very few succeed on their own. They do so in collaboration with others. And the bigger the problem, the tougher the problem, the more you need to consult with other people. And so most scientists appreciate that and collaborate with others who have skills that they don't have, or insights, or apply techniques that they're not familiar with. And it's through that collaboration that real breakthroughs are made.
I see. So what's your guilty pleasure?
Well, my guilty pleasure is watching nonsensical mysteries on TV in the evening and just relaxing and getting away from the day.
I see.
It's just simple entertainment that's not particularly intellectual, but it's just relaxing.
I got you. Who was the person or the individual that had the most influence on you in just being as successful as you are?
Yeah. Well, there are many people that I'd like to credit, but the one who was most influential was my Ph.D. mentor, a man named Arthur Kornberg at Stanford University. As an undergraduate at UCLA, I had developed an interest in how chromosomes duplicate each other. It's called DNA replication. And I had studied it in a certain way, but he was the master of taking the problem apart piece by piece, discovering the molecules that are involved in chromosome replication. And so I was fortunate to be admitted into his program and to be admitted into his lab. And although he was a terribly difficult and tough taskmaster, he expected only the very best from his people and a corresponding crazy work ethic. I learned from that, almost by trial, how to look at a complicated problem and to understand how it could be solved piece by piece. And I did that. I was very successful in his lab and developed a way of doing this using biochemistry, but I knew about genetics. I knew how the two could be combined. And when I started here, I applied that overall logic of his approach, but employed genetics first to kind of bootstrap my way into biochemistry.
I see.
And so, although he himself was very narrow in his focus, he didn't believe in anything other than just isolating molecules using biochemistry. I had a slightly different perspective from what I'd learned before, and I was able to apply the two and make something that was sort of more uniquely my own.
So, in science commonly on TV, we talk about the good stuff, like, oh, this scientific discovery here, this scientific discovery here. And that's great and excellent. But can you explain to the audience here just how ugly things get when you're in the lab and you don't know what to do when you've been working on this project for 2 months and you're stressed out?
Yeah. You have to be very patient and single-minded if you want to be a research scientist. That's like I said, you have to be willing to take risks. And but you have to be methodical and approach a problem step by step and try to figure it out, not in one fell swoop, but in little mini steps. And if you plan right and you think about what went wrong when the last experiment failed, and you try to learn from that failure, then eventually you'll learn something that is meaningful and that allows you to go to second base and then to third base and then eventually to home plate.
What's your advice for the undergraduates or anybody or even somebody that, I don't know, just out there and wants to pursue the field of science?
Yeah. Well, my feeling is, as an undergraduate or even in high school, the best thing you can do is to try to do an individual project, to do something on your own. This is how I got interested in science, by doing annual science fair projects in middle school and in high school. And then when I got to university, I did quite well in a freshman chemistry class, and I was, just by chance, admitted to an honors section where all the students in this particular section, a small subset of the whole class, were asked to pick a laboratory in the chemistry department to work for a term. And for me, that was the opening. My eyes were open to the possibility of not having to go to medical school to apply my interest in biology, but actually to work in an experimental laboratory setting. And that changed my life. And the instructor the young assistant professor whose lab I joined for just 10 weeks, gave me a really very influential book, a new textbook called The Molecular Biology of the Gene, the first edition of a book that was written by James Watson, one of the co-discoverers of the structure of DNA. A man who was very controversial, but who was a brilliant mind, and he wrote this incredible book that talked about life in a way that had not been developed in any textbook that I'd seen. And for me, that book made all the difference. It really turned me on to this subject, and I've embraced it ever since.
And last question for our viewers out there. In the next 5 to 10 years, what developments in biology or in the biomedical realm do you want to see?
Well, I often get asked to predict the future, but I have no crystal ball. And you can answer that question best by looking back at just the past few years to see which discoveries have made a difference that you wouldn't have predicted. So rather than answering your question, I'll answer it by looking backward. One of the greatest discoveries that has impacted our life in the world in the past 2 years was a discovery that led to the new mRNA vaccines that most of us have taken. This is a completely new way of developing immunity, and it came from an idea that was from an immigrant woman by the name of Caitlin Carrico, who escaped basically Soviet-era Hungary and came to the U.S. with an idea that somehow messenger RNA, which is a copy of the genes that gets moved into the cytoplasm to make a protein. Her idea was to somehow employ mRNA to make for some therapeutic application. And she struggled. She tried mightily to get grants, to get an academic position, to realize her dream, and she failed miserably. She was eventually fired by the University of Pennsylvania, where she had a temporary position. But she then had a bench in a lab with a colleague who had security of employment. And they basically figured out how to make mRNA work by injection into the body. The mRNA experiments that had done previously, when mRNA is injected into an animal, it causes a terrible immune reaction that couldn't then be used therapeutically. And she figured out that by simple chemical modification of the RNA, you could make it resistant to immune surveillance. And that key discovery was published in 2005, and it was known but not appreciated until the pandemic hit. And she was by then working at a company in Germany called BioNTech, which is now one of the great achievements in the world, having worked for Pfizer to make a vaccine that many of us have used. A company in the US called Moderna used the same approach, and it was all based on her discovery published in 2005, which has now saved hundreds of millions, possibly billions of lives around the world. Now, her discovery at the time was not seen as particularly special. She tried to get it published in these fancy journals, Science and Nature. Her paper was rejected because they didn't see the value of it. It was published in a smaller journal, but we now appreciate how fundamental that discovery was in a way that no one could have predicted. So I think that can be said about many of the great breakthroughs. Some of them could have been predicted, but many of them could never have been predicted, and so I can't tell you what to expect in the next 5 or 10 years, but all I can tell you is there surely will be discoveries of that sort coming up. In many fields. You know, understanding the brain is obviously a place where a lot of people are focusing their interest, but who knows where we'll find the next great discovery.
Who knows, who knows. It'll be great, though. It'll be great.
There will be great achievements.
Thank you. All right, well, Dr. Scheckman, I do appreciate you. Thank you for your time.
Sure.
Yeah.
Sure.